Welcome back and welcome to the last portion of the RT-PCR section of our lab. This week we will be concluding the wet lab components of our RT-PCR section along with finally analyzing the expression levels of our genes of interest following zebrafish larva exposure to our environmental toxin. So just as a reminder, before we get to lab this week, the first manuscript checkup and the first peer evals are due in the following weeks. Please be sure to upload these assignments on time and don't forget about them. So today is the light at the end of the tunnel, right? This has been a long and arduous journey, roughly 5 or 6 weeks worth of effort, all to finally be allowed to analyze the expression of our genes of interest. Today's activity will essentially be loading gels and imaging those gels, and you will be allowed time to quantify the signal from our DNA products. So on each bench, either your samples are there already, or your TAs and your instructor will bring around the reactions that we set up last week that have been allowed to cycle in our thermocyclers for roughly 35 times. So again, either when you get to lab, your DNA products from our PCR reactions will be on your lab benches, or your TAs or your instructor will bring them around. So each of these tubes should only contain, in a perfect world, if everything worked correctly, each tube should only contain amplified DNA from our gene of interest for each sample, right? If you left your tubes in strips, like I recommended last week, you should have one strip of 4 tubes, and in each tube should only be the amplified gene of interest, if all went well. Sometimes with novel primers, though, this doesn't occur, or sometimes if we had contaminating genomic DNA, we might see amplification of things we don't want. We won't know until we image, and ultimately it will be up to you and discussions you have with your instructor to figure out what exactly is in each of your lanes. So you should have one tube for each sample and gene. So this means, again, same math as last week. You should have 4 samples, right? Your no cDNA control, your no toxin control, and then drug replicate 1 and drug replicate 2. So 4 total samples times however many genes you examined, and don't forget your housekeeping gene. So when we finally image our gels, just as a reminder that our DNA fragments or nucleotide segments will separate out by size. So the smaller they are, the further down the gel they will travel, and the larger they are, the closer towards the top of our gel or wells is where you'll find these big, large segments, right? And this is due to the fact that DNA is primarily negative due to the negative phosphate backbone. So this negative these overall negative molecules will travel towards the anode, right, of our electric field. So recall, anions travel towards the anode in an electrical field. So our DNA segments will be running from positive to negative on our gel boxes this week. And so just to simply, rewind and call this back to our original RT-PCR overview video and discussion. So what occurred last week was we set up our reactions. So we mixed up our master mixes, which contained our selective primers to only amplify our genes of interest. And your groups should have handed these to your instructors. So these tubes or strips of tubes containing DNA from your samples, and again, reagents from your master mix, so that would include nucleotides, our Taq polymerase, and primers. And what happened after you left was I set up our thermocyclers to cycle through 35 times, and that would allow our DNA to amplify 2 to the 35th power times, right? Every cycle, they double. So there should be a whole lot of DNA from our genes of interest present this week, or amplified cDNA segments from our genes of interest this week. So where that puts us for this week, we will be loading our wells. This is meant to be a miniature representation of that, we're going to load our samples into our wells, 1 sample per well. Once everything's all loaded up and our DNA ladder is also added, this guy's already separated out, he's not going to animate out. But once we've loaded all of our wells together, we will start our electrophoresis and we will see separation of DNA fragments. So in this example, we want our amplified DNA product to consist of 125 nucleotides, right? And so we have a DNA band here that's about medium brightness, a faint DNA band here, and then a very bright DNA band here. And then in sample 4, if you want to call it, up here, we seem to have a DNA product that is 500 nucleotides large or long. This would indicate that an incorrect product was amplified and something went wrong during the process. In our lab, it will be necessary to denote that this is believed to be incorrect. In a real lab, you may go through the process of trying to figure out exactly what happened, or you may go all the way back to the starting point and redesign your primers. So, what we can tell from our bands here, our band brightness, remember the brightness of our bands correlate to how much cDNA or DNA is present. So of these 3 bands, the brightest band here would mean there is the most DNA from our gene of interest present. However, this doesn't necessarily mean that our ethanol exposure affected, in this case, increased gene expression, right? That isn't necessarily what it means. If you recall from previous discussions, we need to compare this signal first to our housekeeper gene, right? Which will normalize gene expression of our gene of interest to our housekeeping gene, which is supposed to be unaffected by our environmental toxin. So what this may look like, let's run our gels again. So we run our gels, and now let's compare our signal to our housekeeper gene. So over here is our housekeeper gene. What you'll be doing after we run our gels, image our gels, and analyze the brightness of our bands, which will involve literally quantifying the intensity of pixels from each band, which, again, all that really means is that the brighter the band, the more DNA present, right? Intensity of pixels basically just means how bright our pixels are. So we'll quantify that. And so for an example, we have here, right? So this would be our gene of interest. We're going to say this band has a brightness of 2, and then the brightness of our housekeeping gene only has a brightness, has a brightness of 4, right? We'll analyze the other ones. So recall this is dimmer, so it has a lower score, 1 versus 4, right? The brightness of our housekeeping bands here are identical. And then 4 to 4, and then 0.2 to 1, right? Both of these bands are significantly dimmer than the other ones. And that's why we have to ultimately correlate and normalize the total expression of our gene of interest back to our housekeeping gene. Our housekeeping gene allows us to determine whether or not all gene expression has decreased or just expression of our gene of interest. So when we normalize these out, right, we will see that this comes out to about half, this comes out to about a quarter, this comes out to about 1, and this comes out to about 0.2, which ultimately means that this set of data, 4 versus 4, right, band 3, has the highest gene expression of our gene of interest relative to our housekeeper gene. Now let's look at some images of representative gels to see how this is going to actually look once we quantify and get to Excel. So ultimately, this is what our gels and our bands are going to look like. It is your responsibility to mark up your gels, right? So in lane 1, the DNA ladder should have been loaded. So our DNA ladder is basically a compilation of nucleotide fragments of set or known nucleotide lengths, which correspond to a vendor assigned known series. You will be given this. It is uploaded to Brightspace as well. You will have to mark each of these bands and tell us how many nucleotides are in each band. That will help us resolve how many nucleotides are contained in these bands of our real samples over here. Right? You also need to mark up what is in each lane, right? I know this is the DNA ladder. It's labeled DNA ladder. If these were unlabeled, I wouldn't know which samples are in which lane. So it's your responsibility to write up a template when you load your wells, and then label up and mark up your gels to notify anyone looking at your data what sample is in what well. So in this well, we have our no cDNA TBP. In this sample lane, we have our control TBP expression. And then let's ignore DMSO for now and go over to our drug replicate 1. So when we quantify these, this band has an intensity of, say, 301,002, right? Our control TBP has 2.7 million, and then our D1 has 1.7 million. And if you notice and or you caught, this band actually shouldn't have any brightness to it, right? So in our no cDNA control, there should have been no cDNA to amplify, be reverse transcribed and then amplify throughout our steps. So if you have a DNA band in your no cDNA lane, perhaps there's an error, or perhaps there's a pipetting error, or perhaps there's contaminating genomic DNA. You could, in this case, you can work with your instructor to figure out what the problem may or may not be. So, again, we've analyzed our bands. We have pixel intensity for each of our housekeeping genes. And then if we go over to here, we're going to pretend that these are gene 1, right, or gene of interest 1. So our no cDNA band here only has a volume intensity of 5,002, much, much, much less than our TBP. And then our control gene 1 has 919,000, and then our drug 1 has 1.7 million. So, ultimately, the way you will analyze this DNA, or these results, excuse me, is again by comparing the relative expression of our genes of interest within each sample to the expression of each housekeeping gene with respect to each sample. So, you will take the pixel intensity from control G1, right? So this data right here, 919,000, and you will divide it by the pixel intensity from your control TBP. And that'll look like 919,000 divided by 2.7 million, which comes out to a ratio of about 0.33, right? So that's literally saying this band is about a third as bright as this band, or in total, there's about a third as much DNA here as there is here. So in addition to this, if you have a DMSO control or a vehicle control, you can perform the next step. We are going to pretend that this is actually our drug treatment, right? So pretend DMSO is drug. So we've analyzed how gene expression in our DMSO divided by gene expression in our TBP DMSO. And that comes out to about 0.66, right? So it's saying this is about 2/3 as bright as this band. So next, we want to compare the relative gene expression of our gene of interest to TBP in our drug-treated group to our control group. So we simply divide our ratio of drug expression by our ratio of our control. That'll look like this, right? DMSO G1 divided by DMSO TBP, and then that divided by control G1 divided by TBP. So 0.66 divided by 0.33, which comes out to 1.98, which ultimately means that treating our larva with DMSO, or our drug, or vehicle control in this case, has resulted in a 2-fold increase of our gene of interest as compared to control, right? So this means giving our zebrafish larva, again, in whichever treatment you want to call this, has increased expression of gene 1 2-fold, which means that causes gene expression to increase. It may decrease, it may stay the same. If you need help working through these results, contact your instructor and they can help you interpret, graph, and label your results for this. There are also steps in the protocol to do so. So these are just some last minute notes. We will be reusing our TAE running buffer. So we will be making TAE agarose gels and running our samples through TAE buffer. Each comb will have 15 lanes. So in total, you could technically label 14 samples per each column, or excuse me, it's really row. But we're going to organize that a little differently. Your instructor will go over that at the beginning of class. One of these lanes will be taken up by the DNA ladder, which your TA or your instructor will load, and then we'll load your samples with leaving a space in between so we can hopefully make sure that nothing gets messed up. When you load your samples this week, only load 18 microliters of your samples, and remember, you don't want to go down to the second pump of your pipette when loading these samples. Simply go to the first stop, which will eject your sample, and then remove your pipette without going to the second stop. If you go to the second stop, you will introduce a small air bubble into the well, which can throw your sample up into the liquid, causing it to come out of the well, which will diminish the signal ultimately when we image our gels. The plan for this is to run our gels at 60 volts for 20 minutes. And again, this is specific to our gene ladder. We'll be using a 1 kilobase pair gene ladder, and information from the vendor is uploaded on our Brightspace page. This is an example of how to organize our wells this week. This is what I meant when I said there's 15 wells per lane, or 15 wells per comb. One of those lanes will be taken up by our DNA ladder. You will leave a space between your DNA ladder and the first gene that you load. Then you'll load all 4 samples, right? Your no cDNA control, your no treatment control, DMSO or drug replicate 1, and then drug replicate 2. Leave a space, load the samples for your second gene of interest, leave a space, load the samples for your third gene of interest, etc., etc. You should load TBP last. Please be sure to load TBP last so that your instructor can help you interpret the image and your results from your gel.